All right, let's get this show on the road. We're going live. 3, 2, 1, maybe.
Hey, what's up?
Is the volume going to be better today?
Yeah, that's what we were praying with the mics. All right, ladies and gentlemen, and hopefully, like I said, the mics are working today. We won't have the issues we had yesterday. If you are waiting online to go or to watch, or you're trying to get the end of it, and I'm sorry about yesterday. I'm happy to post any of the PowerPoints too if you want them, but most of the stuff is what I drew on the board. It's just kind of like blank to give me light and such, and I can't do anything to light my face more, but it is what it is. So today's Bioenergetics, but we're going to start off with doing some review of multiple choice questions the way we did yesterday. And so I picked a couple. We'll spend probably about 5 minutes on this max. So if we don't get to all of them, we can go back and look at them, but I want to start with this. So same thing as you've done before. Go through, make sure you're picking out the important parts. Look at the diagram. Okay, always start with the diagram and see what it is that they are talking about, and then go down to the question. So for this one, it says tyrosine, valine, proline, then it has an arrow, and then it has tyrosine, and it has peptide bond labeled, valine, peptide bond labeled, and proline. Now, I've been doing this for a long time, so I look at that, and I immediately know what tyrosine, valine, and proline are. But maybe you don't. But once you look at what it says on the other side, can you tell me what each of those things are? Amino acids. So I've got an amino acid, plus an amino acid, plus an amino acid, and then it's yielding, because remember this arrow means to yield in a chemical equation. And then what do I have on this side? A protein. So we're literally, what process is this doing?
Protein synthesis.
Yes, protein synthesis, but even more specific, or maybe less specific would be better. If I'm taking these, I have 3 individual, right, because I'm taking a monomer, and a monomer, and a monomer. I'm going through dehydration synthesis, and I'm making a polymer. Okay, so that's basically what it is, and we're labeling it. So now look, and look, that's what the statement says. What statement best describes the event shown in the diagram? So now we're synthesizing something, we're making something. So you just told me it's dehydration synthesis. So immediately I'm going down and seeing synthesize, I'm seeing joining together, I'm seeing broken, and I'm seeing forming. Okay, and here's dehydration synthesis. So those are all those things that make things to me, or makes sense to me based on what you just said. So immediately for me, there's one I'm going to knock out without even reading the rest of it. C, why?
Broken.
It's being broken, and we just said we're putting something together. So I'm not even going to reread that. The next one, okay, now I'll start up here, and I'm saying, okay, we've got glucose. We're not getting glucose. See ya. Amino acid monomers are joining together to form a protein macromolecule. Hey. Lipid, nope. See ya. Pretty easy that answer is going to be B. I want, when you get to the exam, for it to be that easy, that you're getting it that quick. Okay? And Mr. Suter just texted me, so, okay, he said it sounds good today. Good, so you can hear me. All right, so let's move on to the next question. All right. Many plants have a waxy coating on their leaves. Which statement describes the most likely structure and function of the waxy coating? Now, I see wax, and it's funny because I think it was Owen the other day told me that the most common...
It was Matthew.
Oh, it's Matthew. Okay, yeah. Sorry.
Yeah, because it was a very odd answer.
So, wax tells me what? Lipid. So all of a sudden, I'm thinking that if it's a wax, it must be a lipid. So I'm going to go down, and I'm looking at this, and this one says protein, protein, lipid, lipid. Well, I know it's a lipid. I don't even have to read those ones. Now I might just double check myself because I have as much time as I want. So the waxy coating is a lipid, the waxy coating is a lipid. So that part's the same. So is it that it can help absorb more sunlight, or that it can help prevent excess water loss? So then you have to think about where it is. It's on their leaves. And leaves are mainly there for photosynthesis, okay? And which means that when we're dealing with photosynthesis, we're also dealing with the process of transpiration. And transpiration is basic evaporation from the leaves instead of from the ground. That's transpiration. So if we have a waxy coating that's on a leaf, what's its purpose? Is a waxy coating going to help you absorb more sunlight? No. What in the leaf is going to help you absorb more sunlight? More chloroplast, more chlorophyll. Okay? Whereas the waxy coating, the lipid, is going to prevent water loss. Does that make sense? All right. Let's see if we can get one more in. This should be easy. So, big word here, enzyme. Okay, so it's asking which graph dash shows how enzyme activity changes as the temperature is adjusted above and below the enzyme's optimum temperature. So you've got enzyme activity on the y-axis, you've got optimum temperature on the x-axis, and particularly right at this location. So what do you know about the optimum temperature?
Is the temperature where an enzyme works best?
It's a temperature where the enzyme works best. So which one of these graphs have the highest point at the optimum temperature? D. Okay, you can see that this is the optimum temperature, and then moving away from the optimum temperature, enzyme activity decreases. This should be like a 5-second question once you look at it. Okay, the other ones, this is saying the optimum temperature is the lowest. This is saying it's right in the middle. This one's saying, again, it's also right in the middle. None of those make sense as far as enzymes. And I know a lot of you left yesterday, but when dealing with enzymes, remember I told you it's like the little old ladies at the mall wrapping presents. Okay? If you have the optimal speed for them and the optimal amount of presents, if you lose little old ladies, you're going to lose that. If you're going to, you know what I mean? And you can't necessarily just increase those if you're not increasing the substrate as well. Okay? So that's D. We're going to do this last one, just because I think this is important, because it's showing you the structure. Okay, so I'm going to give you a second to read this one on your own. So, it tells you that cellulose is a carbohydrate, which hopefully you know, and is a polymer of glucose. Which statement best describes how cellulose is formed within living organisms? So if I were going to change this question, for me, I see the word polymer here, and it tells me that cellulose is a polymer, and maybe I don't remember what cellulose is. So what I would do is I would go and say, which statement best describes how a polymer is formed within living organisms? That's all the question's asking you. It's asking you in a specific manner using a specific macromolecule, but it's just asking, do you know how a polymer is formed? And earlier you told me it's formed through, through dehydration synthesis. And if you think back to that other question, because remember, you can turn to anywhere within the module you're working and look, we thought of an example yesterday where it was something looking like this. Terrible drawing, I'm sorry. To get that. So, is it assembled by bonding individual atoms? It is constructed by connecting smaller monomer subunits. Okay, monomer polymer. All right. It is the product of the decomposition of a much larger molecule. Does that make sense? No, it's asking how it was formed and put together. See ya. It is the result of a physical change that alters the shape of a compound. Does this have anything to do with shape? Okay, so then all of a sudden you've got that one. And again, you see polymer, you see monomer. Clue into the pieces that you know and not just trying to figure out everything if you don't know what some of those words are. One of my students today, and they're not here today, but they specifically asked me about vocabulary. And they said, I feel like I would know some of the answers more if I understood every single vocabulary word that's up there. And I said back, I said, well, you can study vocabulary. There's plenty of Quizlet things that we've posted. You can also go on to Quizlet and just look up and say Keystone Biology vocabulary. And there's probably 20 million different sets that you could go through and flip through if that makes you feel more comfortable. But I know at least with my students, I've been telling you all year to look at the things that you know and break down the words that you do know. So when it's telling you something, even if it doesn't, if it just said, cellulose is a polymer of glucose, you know that glucose is a monomer, you know it's a sugar, like break it down into things you know. Even if it didn't tell you that, look at this picture. What shape is this picture?
Hexagonal.
The hexagonal. And we know that that's the basic shape of our carbohydrate because we drew that yesterday on our little chart. All right, so let's go today, I'm going to skip that for now, into bioenergetics. Okay, bioenergetics. The big, scary word that for some reason is the scariest unit, even though I think it's the most straightforward. Bioenergetics. What does it mean? Okay, what words do we know in it?
Bioenergetics.
Wonderful, look at that, bio. Bio means what? Life. Life for living things. Any other things you see in there?
Energetics.
Energetics. Let's break that down into
Energy.
energy. So bioenergetics is just the energy of life, right? Which means absolutely, I say energy, you say ATP. It's all about ATP. Now here's the thing about ATP. ATP, is this how we store energy? Is that how we take energy and we give it to something else? Like, can I have energy in one cell and then pass it to another cell to use? Can I pass ATP around my body? I use the example with my students as my son's 7 years old, okay, about taking him to, they don't have Toys R Us anymore, really sad, but I'm going to use Toys R Us as an example. Okay, taking my son to Toys R Us and I'm giving him $20. It is like $1 million to him. How quickly is he spending that money? See ya. Is he going to have any change left over?
Oh, no.
No, not at all. I gave him $10 to take to the book fair, and he got $3 from the PTO, so he had $13. He figured it out quick in his head that he could get a pen and 2 books and be good to go. No cents left. Brought nothing home. Got a bigger book just so he could spend the extra 50 cents instead of the one he originally picked out. There was no way he was bringing any money home to me.
$3 for the PTO? What the heck? We didn't get that.
Yeah.
Yeah, we didn't get that either.
Sorry.
Never got anything.
Oh. I thought.
Yeah, he does not go here, but it's just, sorry. So that's like ATP though. You're not saving it, you're spending it right away. Absolutely not saving it. ATP is that quick, must be used, and it's not that it's unstable, but we use the word spring-loaded to describe it. So I think about like there's a couple. I think of a pinball machine, but you can also think, I guess if it's getting warmer, like a super soaker. If you've ever had the super soakers, you had to pump up. And you know how like you can feel the pressure as you go, and the more that you pump, the more it's up, and you can't wait to shoot that right in somebody's face?
Yeah.
Okay? So it's like that where you're building up pressure. So when you're dealing with ATP, we know that this means adenosine triphosphate. which means we're dealing with 3 phosphate molecules. So just for the sake of being really quick, I'm going to draw a box that's representing adenosine, even though it's not really true. Okay, and then it's connected to 3 phosphate molecules. So imagine that you have AMP, adenosine monophosphate. So that's like a couple of pumps. Okay, there's a little bit of pressure, there's a little bit of built up energy there, but it's kind of going to be like that squirt that's like, meh. Okay, so then you pump it up a little bit more, and now all of a sudden we go to adenosine diphosphate. Okay, pretty good. But if I pump it up until I can't pump it anymore, and I have all this built up pressure, I've got ATP. But you can't let that just sit with that pressure there. You have to let it out. So ATP happens very, very quickly, and it releases. And as soon as it releases just a little bit, and it releases that one first break, then you go back to ADP, and then we can pump it a little bit more and release it a little bit more. Now, there are times when we make nucleic acids that will actually break at the second P, but most of the time we're only breaking during the first P from ATP to ADP. Okay, so here's the thing, and another reason I drew this. I've seen sample questions before where they've drawn it without this part, like this, and they say, which bond would be broken when ATP goes to ADP? Do you know how many people put 1?
A lot.
What would that be? Would that be ADP?
No.
No. Right. So it has to be here. So like think logically through that. If you see something, you're like, what is this long random thing? Just think about what that means. Okay? So we've got adenosine triphosphate that we're dealing with a lot because we're dealing with the life of energy. There's 2 overall processes that this encompasses. What are they? Photosynthesis and cellular respiration. Now, photosynthesis has how many parts? 2 parts. Cellular respiration has how many? 3. This is true for what type of cell? Eukaryotic, right? Because as soon as we talk about prokaryotic, we don't talk about all these things. For some reason, and I mean I do it too, but I really thought about starting somewhere different. Every time we talk about bioenergetics, we always start with photosynthesis. And why do we lean towards that?
Because cellular respiration is based off photosynthesis itself.
Because all energy comes from the,
It's not all
okay? So,
humans.
true, but it's, remember, energy's not really created, so it's really a transference. Whoa, no. I moved the computer and I,
Aww.
all right, I'm gonna unplug and I'm gonna plug back in again. Maybe? All right, well, I don't really need it at this point, but it's just, we'll see if it comes, ooh, came back, hurrah! Okay, so what I'm gonna do just to change things up is I'm actually going to start with cellular respiration instead of starting with photosynthesis, because why not? Okay, so when we talk about these processes, though, I want to write down the steps, and then we're going to go back and look at each one individually. So photosynthesis, we said, have 2 steps. Photosynthesis has
Like independent reactions. Like independent reactions.
light dependent and light independent. Or this is also known as the Calvin cycle. Okay, cellular respiration has 3 parts. We have glycolysis,
Krebs cycle electron transport chain.
Krebs cycle. This is also known as the citric acid cycle. called a citric acid cycle because it starts with citric acid. And then the last one is the electron transport chain. And if you want to get fancy, it's actually also called oxidative phosphorylation. We'll save that for an AP lesson. So I drew a line underneath of glycolysis for a very specific reason. Why is that?
Because without oxygen, it just stops. Oh wait, actually, prokaryotic cells only do glycolysis.
Prokaryotic cells only do glycolysis, and without oxygen, it doesn't go any further.
Oh.
Both parts are right.
I thought it was a trick question.
There's so many reasons why glycolysis is important. One, it's the process that any organism does because it happens in the cytoplasm. And we know all cells have cytoplasm. So the separation here is because it happens in the cytoplasm, it doesn't need oxygen, okay? And then we have the Krebs cycle and the electron transport chain that this has to be eukaryotic because this takes place inside of our membrane-bound organelle, the mitochondria. Okay? So that's, when I write the steps down, by drawing a line in my mind, I remember all the different separation from that then. Light-dependent and light-independent, or the Calvin cycle, we're always drawn to that picture that I'm going to draw again today of the chloroplast because we can't help it. But it does help to distinguish. But like I said, I want to take kind of a different route, and I want to start with cellular respiration. So I'm going to keep in mind these steps, okay? And we're going to start, of course, with glycolysis. Okay? Now, oh wait, I'm going to go back one step. What is the point of cellular respiration? What's the goal of cellular respiration? To make ATP. So the goal is to make ATP as much as possible so that we can use it. So goal is to make ATP. Who does cellular respiration? Okay, that's organelle.
Eukaryotic cell.
Only eukaryotic?
All cells.
All cells, because we can just do the glycolysis section,
Yes.
right? But what kind of organisms? Plant and animal. For some reason, it's always written in this world of view that plants do photosynthesis for us. I even saw that last night on Mr. Suter's thing. Somebody put cellular respiration or plants photosynthesize for us. No, plants photosynthesize for themselves. We just benefit.
Yeah.
We benefit from it. They do both sides. A plant does not need us to survive. We cannot survive without having something else transform energy for us. So the goal of cellular respiration, though, is to make ATP. All right, so let's start with glycolysis then. So glycolysis, that word glycolysis, broken down into two parts. We've got glyco and we've got lysis. Glyco comes from the word glycogen. Or we can also say glucose. And we know glycogen is just the way that the polymer is stored. And lysis means to separate, to break. Think of the word hydrolysis. Okay, break with water. Okay, so glycolysis is to separate glucose. So in the cell, on the big old cell, okay, and I'm gonna, here's the nucleus. I know you all want that and all this other jazz that is there. But we're gonna pay attention to the cytoplasm. Okay, so this can happen anywhere. Why is the cytoplasm a good place to do glycolysis?
membrane valve organelles are necessary. But appropriate oxygen nutrients.
There's no oxygen.
Well, there could be oxygen. It doesn't need oxygen, but there could be oxygen. And
Space.
yes, it doesn't have membrane-bound organelles. And sure, there's space, but why the cytoplasm?
Just so
Every cell has it. Okay, great. But what is it made of?
water.
It's like a lot of water, right? And what do we know about water? It's a good conductor. So if we're doing a metabolic process, it's going to allow all of that to happen in a good way. That's why we are aqueous organisms. Okay, so it's going to happen in the cytoplasm. So glycolysis or glucose is a 6-carbon molecule, correct? So a lot of times when we talk about it, we always draw this nice little piece. Now, there's absolutely more to this process than we're going to talk about because this is a very basic level. But basically, what's happening in glycolysis is that you're breaking the 6-carbon molecule down into 3 sets of, like 2 sets of 3. Now I'm going to put this term out there only because I've seen it in a couple of different examples, is that we call these pyruvates or pyruvic acid. So yes, again, there's other stuff happening, but basically glycolysis is happening in the cytoplasm and is the process of breaking glycogen or glucose down into pyruvate or pyruvic acid. Okay, now from that we can then take these other molecules and we can go into the second step, which is the Krebs cycle or the citric acid cycle. It's called the citric acid cycle because what happens is that these pyruvic acid molecules are combined together to go back into a good way to then be broken down again. It's about releasing energy and releasing carbon dioxide and all this jazz. Okay, and so in order this to happen, you need oxygen. Now, do you actually need oxygen for the citric acid cycle? Is there oxygen going into the citric acid cycle? There's not. But the citric acid cycle and the electron transport chain go together, and so you can't have one without the other, and so it just kind of keeps on going. So you need to have oxygen for the end of the electron transport chain. Okay, so now we're going to go into the mitochondria. Okay, so now we're going in the mitochondria. In the mitochondria, we have a couple of different things. We have the outer membrane, we have the inner membrane, and we have the matrix. Okay, so outer membrane, inner membrane, and then the matrix. So what happens is this pyruvate is then going into the matrix. So it's moving into the matrix, and this is where the Krebs cycle will take place. And I should have drawn this bigger, and I didn't. Okay, so step 1, glycolysis. Step 2 is in the matrix. So it's in the mitochondrial matrix. So that's step 2. For this step, basically what's happening is that we are trying to get everything ready for the electron transport chain. The electron transport chain is the show. And the reason for that is, in glycolysis, we're only making 2 ATP. In the Krebs cycle, we're only making 2 ATP. So all prokaryotic cells make are 2 ATP. All that they're making in the Krebs cycle is 2 ATP. We need a lot more than that. And so what happens is that during the electron transport chain, which takes place on the inner mitochondrial membrane, that's when we're going to make, and this is approximate because it can be anywhere from 32 to 36, so we're going to approximate at 34 ATP. That's so much bigger than the other two. So you've got 2 ATP, 2 ATP, and 34 ATP. So when we even all out, the number that they usually use for the entire process of cellular respiration is somewhere between 36 and 38. For our purposes, we've added up to 38. And sometimes actually it can even be as low as 34. But 38 is usually the number you're looking for if they're giving you the question on the keystone. It's 2, 2, and 34. So that's a very, very quick overview of all of that. There's a lot more to get into. For this, we have to deal with NAD+, we have to deal with FADH2, and so anytime that we're dealing with this process, we're releasing hydrogen. And hydrogen, as it's broken off of this, is then given into NAD. So we go from NAD plus to NADH, and we go from FAD plus to FADH2. And so we have a lot of these carrier molecules that are picking up the hydrogens, and they're taking them into the matrix. And then more is given off during, actually FADH2 is made during the Krebs cycle. But all of this is so that we can pump hydrogen molecules across the inner mitochondrial membrane. Why? Because we're creating a waterfall. We're creating a concentration gradient. Once we have a whole lot of hydrogen ions on one side, then we can turn the water wheel. Okay, and the water wheel is an enzyme. Well, I'll make it bigger. It kind of looks like a nose to me. Is ATP synthase. Synthase, okay, if you break that down, synthesizes, okay, ATP, and it's an enzyme. It's an enzyme that synthesizes ATP. And the way that it does that, the way that it makes ATP, is that all of these hydrogen ions that we have been pushing over to this side flood down, and the hydrogens come through, and the hydrogens are met with these oxygen molecules that are waiting. And hydrogen and oxygen make H2O. So then water is released, and as this is churning, okay, it's kind of like, I think about it, like I said, like a water wheel. So as that water wheel is churning, just think also about hydroelectric energy, okay? We churn that waterfall wheel in order to make ATP. And so that process is happening. It has to do with exciting electrons, and like I said, it's much more complex than that. But I tried not to spend too much time going over this and hitting the very, very basics of this. So overall, glycolysis 2 ATP, Krebs cycle 2 ATP, electron transport chain 34 ATP. So now we're sitting at 38 ATP for this process. What else is going in? What's coming out? Because that's really the most important part about knowing cellular respiration. So first thing we said goes in is, okay, so we need oxygen, great, and glucose. So we're starting with glucose, our C6, our H12, our O6. So that's what we're starting with. And we're going to give off at the end water, right? And we're going to give off, as this goes off the screen, carbon dioxide. That's mainly given off during the Krebs cycle. I should have drawn that. And our ATP. Okay. So again, I started with going through cellular respiration because all the time we look at this side of the equation first. We look at the photosynthesis portion. Okay, photosynthesis and cellular respiration are not exact opposites of each other. They're close, but they're not exact opposites of each other. But they are hand in hand. They absolutely do need one another. Okay, so I'm going to go over photosynthesis again very, very quickly to kind of fly through that. So let's talk about photosynthesis. The fun one. The easy one. All of this is really easy, right? No problems?
Yes.
All right, so photosynthesis. We are making something with light. Photo meaning light, which is why we have the process of chemiosynthesis, which is where we're making, I don't think there's an I in there. That looked wrong.
Yes.
I just always say chemiosynthesis. Chemosynthesis, which happens where and does what?
Like the bottom of the ocean.
Okay, so vents and the ocean and uses chemicals. Okay, so this is like bacteria that can't get oxygen. But photosynthesis, okay, starts with light. So we're starting with the big old sun. Okay, so we got the big old sun and it sends down its little UV rays. UVA, UVB, UVC, ultraviolet, all different sorts of stuff coming down from the sun. Okay, and when we get to our plant, we're hitting our big old leaf, right? And so we have our big old leaf and we have our big old chloroplast that everybody wants to see. And I know Mr. Studer went over this in great detail, and so I'm going to kind of go through this very, very quickly, like I said, and briefly. And so when you're dealing with photosynthesis, we have two parts. And I'm going to separate it like this just for the sake of having it separated. But know that when we're talking about chlorophyll and we're talking about where it is in the chloroplast, okay, it's the same way with a cell. The nucleus isn't always directly in the center. It doesn't always look like this picture that we draw. But a lot of times when you see photosynthesis, yes, you see our little stack of thylakoids, okay, our granums there on this side, and the other side they then show you the big old, circle that's happening, and they're like, oh, yeah, so it looks like it's just on like one side. No, this is happening right next to it. It's happening simultaneously. It's all there happening at the same time. But for the sake of, like I said, quickly going through this and reviewing photosynthesis. Okay, so the sun comes in and the sun hits the thylakoid membranes. The discs, the thylakoid discs, kind of think of them like being big solar panels. So like all the chloroplast inside, it's like these big solar panels that are hanging out that are collecting the rays. And what that actually does is it excites electrons. And so these electrons get excited. And they start this whole process. And there's a whole bunch more to it that's occurring. But when this happens, when those electrons get excited because the sun is coming down, it's able to do a couple of different things. Okay, the first of that is that it is taking what? What comes in besides sun? Water. Let's go start with water. But yes, CO2 is also coming in. So we've got water coming in and we've got CO2 coming in. I'm writing it like this mainly because we're not touching CO2 until we get over there. Okay, so we have water coming in. Okay, so we have water coming in, and when the electrons are excited, it breaks the water molecule. Okay, and when the water molecule breaks, that hydrogen comes off and oxygen leaves. Okay, and so you have to have something to pick it up. And so what picks it up is NADP. Now, how do I remember the difference between cellular respiration, NAD, and photosynthesis, NADP? It's the P. P is in photosynthesis. Okay, I'm all about letters and making sense because they're basically the same prospect or job is to pick up hydrogen. So NADP plus comes in, NADPH leaves and goes to the other part of the cycle. ADP plus a P plus a phosphate comes in, okay? Little boy sitting there pumping his little toy. to build up the energy to squirt the water gun, okay, and ATP is going in. But wait a second, we just made ATP, why are we going to spend it? The reason that we're making the ATP is so that we can run the Calvin cycle. But again, why don't we just want to just make ATP? What did I tell you before about ATP? It has to be used right away. So we want to store it. Okay, so we have now the hydrogen coming over with the CO2, which is where we're getting our glucose. So like I said, this is a very, very, very quick rundown of photosynthesis. I didn't want to spend too much time going through it because it's about what's going in and what's going out. And if you understand that, then you should be able to answer a majority of questions on that. Okay? Any further questions about photosynthesis or cellular respiration that you want me to go into? All right. Well, I told you I was going to try to make it quick today. Have a good afternoon.
Thank you.
Hopefully, you can all hear me everywhere, and I will press the little stop button. And since some of you are all here, if I get more snacks tomorrow, do we have a preference?
Giant cookies.
Doritos.
Giant cookies.
So here's the problem with the cookies.